fibrosis, they just started linkage analysis using those markers in the disease state. Which markers seem to travel with those individuals that have cystic fibrosis? Which variant of a marker? So each marker may have a slight difference in the sequence, and so which variants tend to travel, okay? By doing that, they were able to narrow it down to chromosome 7. And so remember, the name of the game is being able to trim down that massive 24,000 gene, 3 billion base pairs of information down to a more manageable size. Ultimately, at the end of any analysis, what you'd like to get is a gene list of, say, a dozen genes. Because now you just took 24,000 genes and cut it down to a dozen. It makes it more of a manageable thesis topic than trying to tackle all 24,000, for instance. Okay, and so they did linkage analysis with these markers and were able to find a region of interest. Okay, and then they did positional cloning. And we talk about positional cloning, remember we digest up our genome and we put it into these plasmids that we call BACs. Okay, a BAC is nothing more than a plasmid for a big piece of the genome. And so we can have all of these different pieces of the genome that we just put into bacteria. They go onto the plate, and so this colony may say be that piece of DNA, that colony may that colony may be that piece of DNA, that colony might be that piece of DNA, that colony might be that piece of DNA, that colony might be that piece of DNA, on and on and on. And we end up, and when you actually do this, it's called a BAC filter. And so for like, I've done screens on the Tamar wallabies, the marsupials BAC filter, and it's a filter about this big, and on it is 9 regions. Each region has, I don't know, probably about maybe 250 spots. Each spot has about 8 spots in it. And so you do this on a massive scale. And then for the whole Tamar genome, it's like 8 of these filters. And so you take your sequence of interest and you start screening it, label it with radiation. And so what they did is they identified 4 genes, or they did positional cloning to identify all the clones to span a region, sequenced all of those clones, and then once they sequenced all those clones, you could just look for open reading frame. Where do you have a start and a stop that's in frame? That's likely a gene, okay? So now you can say, okay, well, here's one gene, here's another gene, here's another gene, here's another gene. We now have 4 genes in this region that could be contributing to cystic fibrosis because it's linked to cystic fibrosis. And so then they took those 4 candidate genes and then they did northern analysis. And so again, this is just looking at differential expression, because the important thing is trying to identify, is using all these pieces of information to reason out what is the most likely gene, okay? They have 4 going into the northern. You take RNA out of each one of these tissues, run it on a gel, and put it onto a membrane. All that putting it onto the membrane is just a more durable way of doing it. A gel falls apart. And so you want to get that stuff out of the gel onto something more permanent, which is a membrane. On the membrane, what you did is you loaded your RNA up here. The wells are probably up here. And then you run it just by gel electrophoresis down. And then these are all from all these different tissues. Then you transfer that to the membrane. You take and make a radioactive probe to each one of these candidate genes. Individually, you take it, put it on your membrane. If that gene is there in the RNA, your probe's going to stick to it. You put X-ray film over your membrane. Wherever you get signal, that means you have that particular gene in that RNA pool. Okay. Now, the significance of this, remember, was pancreas, nasal, lung, and testis were some of the main organs that are affected with cystic fibrosis. So we use linkage analysis to find the region. We identify the 4 candidate genes. And now, which of these 4 candidate genes are the ones that we're interested in? Well, we can use expression analysis to identify which of those 4 genes is actually on in the affected tissue. So, we got all of wherever we see a signal, that's where our probe stuck. It's stuck because it had complementary sequence there, so it hybridized there, and we detected it because of the radiation on the X-ray film. So now we know that this particular gene has the expression profile similar to what we see in the affected tissue of the cystic fibrosis individual. We're then able to then say, okay, well, that's the potential cystic fibrosis gene. So let's sequence that gene, and we can just do this by PCR and Sanger sequencing, like we've studied already. Let's sequence this gene in a whole bunch of people with cystic fibrosis. Let's sequence this gene in individuals that don't have cystic fibrosis. And what they found is that in that particular family, there was a deletion. So we talked about how deletions can affect and how affect protein and all that stuff. And so this is the way we can use these techniques, and all based on those basic molecular principles of complementary base pairing and the way DNA gets synthesized. Now, we also talked about the genome. What did we go to? We go to 11. Right? Yeah. And the way we're able to assemble the genome. And so essentially what the map-based approach is essentially what the researchers for cystic fibrosis did here. Where they identified this region, what they're doing is they're just doing that chromosome walking. They're taking a little piece. They identify one clone, so they have a marker gene. They have a clone, and when we're talking about clones, each one of these is a clone. Okay, and so if they identify a clone, if they have a clone that has their marker gene on it, well, then they can make a little. They can take this sequence here, make a new probe, and identify that clone. They take a sequence at the end of it, make a new probe, identify that clone. Okay. There's another thing called chromosome jumping that we didn't talk about that lets you go further distances. But so you can take this, and then once you get a little further, you can make a probe for that end and identify that clone. Make a probe for that end, identify that clone. Do some chromosome jumping. Here, if you make a probe for that end, you can identify that clone, and you can kind of walk your way through. That's what map-based sequencing is. Okay? It's this stepping, identifying that minimal tiling path, and then sequencing that least amount of context to get, the least amount of clones to get across a region to develop content. Versus the shotgun sequencing, which is blow it all up, sequence all the little pieces, and put it back together. Talked about how these two things were used to assemble the genome. Well, what does the genome do for us? Well, the genome ultimately does a lot of different things. It's kind of the starting point now that we're able to have the genome. What we're, I do a writing class on genomics, and one of the articles we're reading for today actually is it's a piece, I'll actually post it on Husky CT in case you're interested. It's just came out like 2 months ago. It asked like 8 of the top people in the field of genomics, different areas of genomics, what their projections for the next 10 to 15 years are for the field. And what unanimously, what they all pretty much say is that it's gonna impact medical field most significantly, agriculture pretty significantly as well, but we're gonna sequence genomes, and we're gonna sequence a whole lot of genomes. And one person actually predicts that was in the genomics, human genomics field, that within 15 years we'll have over 1 billion genomes sequenced. Okay. And then we'll have over 1 billion genomes sequenced, and then the goal will be with lots of layers of privatization or privacy and personal protection, and that's a big issue when you're talking about this type of genetics. If you went to the Henry Lax thing, that's a big issue is maintaining privacy in this type of data. But if you have 1 billion genomes sequenced, and then you're able to connect that to your medical records, and every time you go to the doctor and you have your blood pressure taken and you have high blood pressure. Blood pressure, it's obviously got a big genetic component, but it's got a lot of environmental components. And if we're able to start tying different phenotypes to different genotypes, is how we're going to be able to start tailoring medicine and start being able to identify these different variants that give us these different disease states. And so we ultimately need a map, and that's where our physical map comes in. We assemble our genome. We start overlaying all of these different pieces of information on the map and start looking at where things co-occur. Where do we get a recruitment of H3K27 acetylation? Okay, acetylation, remember, was promoters, and so we see little peaks where we're going to get in promoter regions, for instance. Okay, different histone modifications. The other thing that we can do with the genome is we can start looking at variation within the population. And so we talked briefly about SNPs, and so SNPs are just single nucleotide polymorphisms, differences between us. And so what we know is certain regions are prone to SNPs. Between most people might have some differences. And so, but those differences are heritable differences. And so whatever set of SNPs you have, that's called the haplotype. Your haplotype you got from one of your parents, okay? Or from both of your parents. You got one haplotype from your mom, one haplotype for your dad. For any particular region of the genome where we know what the SNPs are, we can assess them. We can just design primers bounding that region, amplify it, sequence it, find out what your haplotype is, okay? We can do this on a grand scale in which we simultaneously evaluate thousands of loci in one PCR reaction and one sequencing reaction, in which we amplify all of these different regions with SNPs. We identify all of the SNPs that you have in thousands of regions simultaneously. And then what we do is we do a giant linkage analysis of what phenotypes do you have. Do you have heart disease? Do you have pulmonary disease? Have you had cancer? Okay. What SNPs do you have, and what SNPs, what haplotypes tend to travel with those particular disease states? Okay. And so, in this way, we're going to be able to evaluate and identify lots of very complex diseases, all of that stuff we talked about in quantitative genetics. This is how we're going to be able to narrow down and get to those answers. Now, microarrays, again, this is just another instance of hybridization. All we're doing is using complementary base pairing. We're putting little spots on a slide. On this slide are little tiny spots. Those spots are little pieces of sequence that are complementary to a target gene. And so we take our map, and so here's a gene, SOX5. Each of those, the lines going down, that's an exon. So we take a little sequence from that exon, 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 and we put each one of those on a different spot. And now, complementary to that sequence. You take your RNA, you label it with fluorescence when you turn it into cDNA, stick it onto your slide. Wherever you have spots that are complementary to your RNA, they're going to hybridize by complementary base pairing. Where you have hybridization, that's going to mean you are hybridizing your RNA, that is now cDNA, that has fluorescence on it. And so you're then going to be able to detect the level of fluorescence. The more fluorescence you have, the more RNA you have. Okay? You have two different samples, one red, one green. You put the two together, put it on the slide. It's called a competitive hybridization because they're both competing for those little pieces of complementary sequence on the slide. But some red will stick, some green will stick. If equal amounts stick, it means you have equal expression between your two samples. You have your yellow spot. If in your cancer you have an oncogene that's being way overexpressed and you label that in red, and normal is green, well, green, you're going to have some stick, but you have way overexpression in your cancer sample, and so you're going to have more red stick, you're going to end up with a red spot. That'll probably be like an oncogene. The tumor suppressor genes, it means it's turned off in your cancer. So that means you're going to have more in your healthy tissue, green, than in your disease tissue, your cancer. So more green's going to stick, and that'll give you a green spot. And then the ones with nothing, those are the ones that gene is off in both cell types. Okay. And so again, we're using hybridization to assess this global gene expression that we're able to simultaneously ask, what is the gene expression of these thousands of genes, and how do they differ between two different samples? So last thing to say is next generation sequencing has totally revolutionized the field. We now are able to sequence the entire sample rather than pick little tiny pieces and sequence these little disparate kind of random pieces. We're now able to sequence the entire thing, the entire genome, the entire transcriptome, whatever. All the small RNAs, all the mRNAs. And this really has revolutionized the field. It's created lots and lots of data. It ultimately will get replaced because all of next generation sequencing platforms right now sequence on a really, really little snippets, 50 to 100 nucleotides on average. Some go up to 400 or so. But the next round that's coming out out of England, what's the name of the company? Is it ION? No, it's not. No, it's I forget the name of the company. It's a new company that's coming out. They're doing long reads where they have a pore that the DNA gets pulled through and sequenced as it gets pulled through the pore, this nanopore. And as it gets pulled through, it gets sequenced, and they're able to sequence as much as 50,000 to 100,000 base pairs all at once. And what this does is rather than blowing up the genome and having to assemble all these little tiny pieces, it takes away that assembly process because that assembly process has errors. So next generation sequence revolutionized the field, but will soon be replaced. So, if you don't have any questions, thank you for your attention. Hope you guys learned something this semester. How should
we best study for
the final? You're going to want to review your study guides to kind of get to make sure any terms you're not real familiar with. You're going to want to use the practice exams and your discussion material is probably your best bet to focus in on stuff. Discussion material, the practice exam, and then go over the study guides to identify anything that you like. what was that? You're probably gonna wanna go at least refresh it.